Welcome to Developmental Biology. Today, we are going to explore one of the most critical stages in early embryonic development, the blastula. Imagine taking a single block of clay and slicing it into hundreds of tiny pieces without adding any more clay. That is exactly what happens right after fertilization. We will discover how this microscopic structure forms the essential scaffolding for all future organs and why its shape varies so dramatically across different animals. Now that we know what we are studying, let us look at how it begins. Right after fertilization, the single-celled zygote undergoes a rapid series of cell divisions known as cleavage. What is truly fascinating here is that the embryo does not actually grow in total size during this phase. Instead, that original massive cell is simply carved up into many smaller, manageable cells, which we call blastomeres. This rapid multiplication is essential because it provides the building blocks needed to construct the complex architecture of the developing embryo. Building on this process of cleavage, the embryo eventually reaches a stage called the blastula. You can think of the blastula as the foundational scaffolding of a building. It marks the end of rapid cell division. In its most classic form, these blastomeres arrange themselves into a layer that surrounds a fluid-filled central cavity known as the blastocele. This specific architecture is not just for show, it is absolutely critical. The shape and structure of the blastula dictate exactly how the embryo will fold inward during the next major developmental phase, which is called gastrulation. Understanding the basic definition, let us now explore the different architectural styles, starting with the most classic form, the coeloblastula. If you picture a hollow rubber ball, you know exactly what a coeloblastula looks like. The blastomeres form a single, neat layer on the outside, leaving a large, prominent, fluid-filled cavity, the blastocele, right in the center. This beautiful, symmetrical structure is commonly seen in amphibians, like frogs, as well as marine organisms, like sea urchins. The large cavity provides ample physical space for cells to migrate later on. In stark contrast to the hollow rubber ball we just saw, we have the stereoblastula. Imagine a solid baseball instead. In this variation, the cells divide, but they do not secrete fluid to create that central cavity. The result is a dense, completely solid mass of cells. Why does this happen? It is entirely due to the specific amount and distribution of yolk present in the original egg, which alters how the cells pack together. We typically find this solid architectural style in certain types of worms and mollusks. Moving on from solid balls, what happens when an egg is packed with a massive amount of yolk? This brings us to the discoblastula. Think of a chicken egg. The yolk is so thick and dense that the dividing cells simply cannot penetrate it. Instead of forming a ball, the cells form a tiny, flat disc that sits right on top of the giant yolk sphere. It looks much like a small frisbee resting on a large beach ball. The yolk remains uncleaved, serving purely as a massive nutrient reserve for the developing embryo. While birds have their flat discs, insects like fruit flies have evolved a completely different and fascinating variation called the periblastula. In this case, the cells do not actually divide completely at the very beginning. Instead, the nuclei multiply rapidly inside the egg and then migrate outward to the very edge. Once there, they form a superficial single layer of cells that wraps entirely around a central mass of uncleaved yolk. It is a highly specialized adaptation that allows insects to develop rapidly while keeping their nutrient supply safely enclosed in the center. Finally, we arrive at the blastocyst, which is the unique blastula form found in mammals, including us humans. Unlike the simple hollow ball of the frog, the blastocyst is highly specialized and has two distinct parts. First, there is an outer ring of cells called the trophoblast, which will eventually form the placenta. Second, pushed to one side inside the cavity is a dense cluster called the inner cell mass. This inner mass is incredibly important because it is the exact group of cells that will develop into the actual baby. This structure is an evolutionary marvel, perfectly adapted for implanting into a mother's uterus rather than developing inside a hard eggshell. Now that we've discussed these variations, let us look at them side by side in this diagram. In the top left, you see the coeloblastula, clearly showing that large, empty central cavity, the blastocele, surrounded by a single layer of cells. To its right is the stereoblastula, which is entirely filled with tightly packed cells, lacking any cavity. In the bottom left, the discoblastula illustrates how the tiny cell disc rests on top of a massive, uncleaved yolk. Finally, in the bottom right, the mammalian blastocyst highlights its two distinct regions, the outer trophoblast ring and the crucial inner cell mass tucked inside. Notice how the physical constraints of yolk and evolutionary needs dictate each unique shape. Seeing these different shapes naturally leads to an important question. Why do we care so much about the exact architecture of a microscopic ball of cells? The answer is that this shape is the blueprint for all future organs. If the blastula does not form its cavity correctly, cells will not have the physical space they need to migrate and form the gut, nervous system, and muscles. In human medicine, this is critical during in vitro fertilization, or IVF. Embryologists carefully study the blastocyst under a microscope to evaluate its health and structural integrity. They must ensure they are implanting a perfectly formed, viable embryo that can successfully attach to the mother's uterus. To understand how we get from a single fertilized egg to a complex blastula, we must look at the mathematics of cell division. During cleavage, cells divide exponentially. This means the population doubles with every cycle. The formula governing this is n equals n0 times 2 to the power of n. Let us break down these symbols. N represents the final total number of cells in the blastula. N0 is our starting point, the initial number of cells, which is exactly 1 for a single fertilized egg. Finally, N represents the number of cleavage cycles that have occurred. As the number of divisions, N, increases, the total cell count, N, grows incredibly fast, providing the thousands of building blocks needed. Now that we have our formula, let us see it in action with a concrete example. Imagine we are observing a frog embryo in a pond. We are given that the single fertilized egg has gone through exactly 12 rounds of cell division. We need to find the total population of blastomeres. We start by identifying our knowns. N0 is 1 and N is 12. We choose our exponential growth formula because cleavage doubles the cell count each round. Substituting our values, we get N equals 1 times 2 to the power of 12. Calculating 2 to the 12th power gives us our result. The final answer is 4,096 cells. Physically, this means the embryo has rapidly multiplied to create enough cellular building blocks to begin shaping the complex tissues of a tadpole. Let us try one more example, this time working backward in a clinical setting. An embryologist examines a human blastocyst and estimates it contains approximately 128 cells. We want to know how many rounds of cleavage it underwent. We are given the final count, n equals 128, and the initial count, n0 equals 1. We set up our equation, 128 equals 1 times 2 to the power of n. To solve for the unknown exponent n, we need to express 128 as a power of 2. By recognizing our powers of 2, we know that 2 multiplied by itself 7 times is 128. So 2 to the 7th equals 2 to the n. Since the bases are the same, we drop them, revealing that n equals 7. This tells the doctor the embryo has successfully completed 7 synchronized division cycles, indicating healthy development. We have covered a lot of ground today, so let us review the key takeaways. First, the blastula is a crucial transitional stage that transforms a single-celled zygote into a complex, multicellular architecture. Second, we learned that this shape is not universal. Whether it is a hollow coloblastula, a solid stereoblastula, a flat discoblastula, or a mammalian blastocyst, the structure is perfectly adapted to the organism's yolk content and evolutionary needs. Third, this early architecture is vital because it dictates the entire future body plan and allows for cell migration. Finally, we saw how simple exponential mathematics allows us to track and predict this incredible cellular growth. You now have a solid understanding of the blastula stage,